Hello, my name is Peter Klapper. I'm a senior lecturer in biochemistry in the School of Biosciences at the University of Kent in Canterbury, United Kingdom. In this screencast, I want to tell you a little bit about the research interests and the research questions of my group. So, one thing that we want to find out is how do proteins actually fold? And folding is a very complicated process, and we already know that there are so-called molecular chaperones. These are other proteins in the cell that assist a protein in the folding process, that make sure that a protein can reach its functional three-dimensional state. And we want to know how do these molecular chaperones actually do that. We also want to know how these molecular chaperones interact with their substrates on a molecular level. So, for example, here is a typical molecular chaperone, a so-called peptidyl prolyl cis trans isomerase. We've got the structure of this protein. And we want to know how this protein actually interacts with other proteins to help them to get to the right state. Here we see a bound substrate protein, which interacts closely with this molecular chaperone. And we want to understand how this is actually happening. So we try to identify the amino acid residues of the substrate protein, here these the colored amino acids that interact with the certain amino acid residues in the molecular chaperone. Why do we need this information? Why is this important to understand? We know that some molecular chaperones are important for the infection with the HIV virus. Without these molecular chaperones, the HIV agent would not be able to infect a cell. We also know that other molecular chaperones are important for the infection with Salmonella and E. coli. Without these molecular chaperones, these microorganisms could not be pathogenic. It is therefore very important to understand how these molecular chaperones actually work. And a good understanding of these mechanisms will allow us to develop new and better drugs against these diseases. So hopefully this video gives you a little bit of insight into what the research questions of my research group are, and thank you very much for watching it.